Andate a casa, non c'è nulla da fare. 25 anni fa, una diagnosi di distrofia muscolare di Duchenne.
È vero che i miei muscoli diventano sempre più deboli.
Una patologia degenerativa rara che colpisce i muscoli. Ma c'è chi non si arrende e fonda Parent Project. Intanto il mondo cambia e anche Parent Project cambia un po' il mondo.
Ringrazio Parent Project e
l'ultimo anno finanziato da Parent Project.
Parent Project che ha studiare in maniera...
I pazienti oggi hanno un futuro da immaginare, degli obiettivi da raggiungere e dei traguardi da conquistare. Oggi, anche con la Duchenne puoi diventare ciò che vuoi. Come Luca
Buzzella, che ringrazio per essere qui.
Morire insieme, in fin dei conti, è una sfida già vinta.
I'm here to tell the future is there.
Jacopo,
Alla Duchenne.
Matteo, Lorenzo, Luca, Samuele, Francesco e come tanti altri ragazzi che vivono una vita piena. Ma questo non basta, non c'è ancora una cura e tanti sono gli ostacoli da superare e le strade da aprire.
Tutti insieme abbiamo formato una grande famiglia.
Scrivi i prossimi 25 anni di storia insieme a noi.
Sulla nostra navicella spaziale.
Welcome on our spaceship. With this, we're about to kick off the first scientific session of the 19th edition of the International Conference on Duchenne and Becker Muscular Dystrophy. Parent Project's journey never stopped over the last 25 years. And this year, we're all looking forward to meet again in person. And we've decided to hop on a spaceship, looking for new ways to keep traveling towards our common goal. In order to organize this event, it took us months and months of work, and we'd like to particularly thank our staff, the Parent Project staff, and the people you can see on the screen right now. I'll be seeing them in the next few days. You saw yesterday's, and even the people that you can't see that are working backstage. I'd like to thank all speakers for joining us from all over the world over these days to provide us with scientific and non-scientific updates on our disease. Thank you all very much for making it possible to travel on the spaceship. I would like to thank Carlo and our technical team. A particularly warm thank our sponsors, PTC Therapeutics and Ital Pharma, and our platinum sponsors, Sarepta Therapeutics. Thank you very much for the support given to our international conference. And we'd like to thank all the other companies that supported our event and made it possible for us to organize it. I'd like to particularly thank our interpreters, which they thank you back, Gabriella, Simonetta, Vanna, Valeria, and Paula. Thanks to them, this event can be truly international. And we should all bear in mind to please speak slowly to help them do their best to translate every single word you say in the language you choose to listen to. In order to choose your language, just click on the globe icon and you then can select the language you want to listen to, Italian or English. The conference is being streamed on the Facebook page of Parent Project and on our YouTube channel on Facebook in Italian and on YouTube in English. If you want to follow all sessions, just visit our website. Just click under the titles of each session and you can attend the live streaming in that way. On the website, you'll be able to immediately take a look at all the posters sent by our researchers. And tomorrow, Saturday, February 19th at 12 o'clock, we'll go live with all the authors. So you'll be able to ask your questions on all posters. On our blog, Paola Nespoli wrote, "The Earth is a ship or a spaceship that we all travel together on. We're not traveling as tourists, but as crew. And in fact, the only way for the ship to keep traveling is for the crew to keep rowing all together towards a common goal." And it is with these words and a renewed wish for all of us to enjoy the conference that I'm going to give the floor to Ilaria Zito, a biologist from our science area that many of you already know, and Luca Genovese, the president of Parent Project, who keeps steering us every day from planet Earth. And let's open our session on scientific update with a perfect title. The title is A Glimpse at the Research Horizon on Duchenne Muscular Dystrophy. Then we'll have the parallel sessions to be introduced by Ilaria before the end of the session at 5:30 p.m. Over to you, Ilaria. So Ilaria, and you on the spaceship, and Luca on the Earth. You're in charge of the session. Thank you very much, Christina, for this introduction. I'm really excited to be here again. Of course, virtually again, not in person, and chairing the 19th conference on Duchenne and Becker Muscular Dystrophy, hopefully the last one held remotely. Let's start our first scientific session of the conference. As mentioned by Christina, the title is extremely insightful, A Glimpse at the Research Horizon on Duchenne and Becker Muscular Dystrophy. It is a cross-cutting session which goes hand in hand with the slogan of the conference that you've probably seen already, a journey towards the future, drawing new maps. During the session, we'll get an update on a number of interesting topics, from genomic editing, which is a strategy that we've known for a number of years already. There are great expectations by the entire Duchenne and Becker community to finally start transitioning from preclinical to clinical setting. But there are more recent studies as well into new and more advanced alternatives, the so-called new generation strategies in order to make treatments more effective. There's a tremendous activity in the scientific community, with people constantly striving to draw the new maps mentioned in our slogan that will hopefully characterize a not too far future. I will immediately give the floor to our president, who is here with us and who will certainly provide his own introduction, and he will personally introduce the different speakers of the session, and I'll be back with you later on. Thank you very much, Ilaria. Let me say hello to all of you. I'd like to thank all the speakers of the session. They're ready to take the floor, an especially warm welcome to all attendees. my capacity as president of Parent Project and as the father of 2 adult Duchenne sons, I do have great hopes in scientific research. I trust the scientific research. I've always followed the scientific research over the years. I've taken part personally in 13 conferences plus 2 held remotely. So I can say I got some experience there, and my emotion is always extremely high. I feel always excited. When Duchenne entered our lives, it was a breakthrough. It set a new beginning. I've been following all the progress that has been made over the years. We sometimes had the impression that things were not taking off and we were stuck, but little by little, something's been achieved. And in fact, the first advanced therapy experimented on our children will be described tomorrow in the session on gene therapy. So we're still very confident, truly confident. We should never lose our confidence, and we listen to and follow whatever the scientific community tells us about recent progress in research. So, and we're all extremely focused on our final goal, to finally defeat this disease. It is my great pleasure now to immediately introduce our first speaker that I'd like to thank very much for joining us to start with. Our first speaker will talk about an extremely important and new topic, genomic editing. So we have one of the top international experts in the field, Charles Gersbach, a professor of biomedical engineering at Duke's University. Let me immediately give the floor, and thank you very much for joining us today. Over to you, Charles.
Thank you so much for the kind introduction and the opportunity to come and present some of our work on genome editing today. I greatly appreciate the opportunity to speak, and wish I could be with all of you in person, but maybe sometime soon, hopefully that will be possible. The work I'm going to talk about today is just a quick update on where the field is on applying genome editing to muscular dystrophy. And I'll first show my disclosures, but really the important one here is that our work in the area of gene editing for Duchenne for the last couple of years has been part of a fantastic partnership with Sarepta Therapeutics, and we greatly appreciate all of their support for our work. As quick background to gene editing as a transformative technology in the genetic medicine area, what's unique about gene editing is really the ability to go in and modify DNA sequences where they exist naturally in the chromosomal DNA. So this is in contrast to other types of modalities being applied to Duchenne muscular dystrophy, such as RNA-mediated approaches using antisense oligonucleotides that induce exon skipping at the RNA level that need to be continuously re-administered as the RNA is degraded and turns over. And it's different than the microdystrophin gene transfer strategies that are adding an extra copy of of a microdystrophin gene to these cells. In contrast to those approaches, we are manipulating or changing the DNA sequence of the dystrophin gene in its natural position in the chromosomal DNA. And there's a couple of different potential advantages of this approach of gene editing to correct the dystrophin gene in its natural position. And many of those are listed here. And for time, I won't go through all of them, but I'll highlight that by correcting the gene in its normal position, we get the natural levels of dystrophin being produced. There's also the potential, because the gene isn't already encoded in the genome, to create a larger gene than what is delivered in the microdystrophin constructs, which may potentially have increased levels of function. And then additionally, for the approaches that involve removing exons and reframing the gene in the DNA, there's no introduction of new protein material that's at the dystrophin, in the dystrophin protein that could potentially be immunogenic. So another question would be, if we're manipulating the DNA sequence of the dystrophin gene, what are the types of manipulations that we're doing? And a general characteristic of genome editing is that it's easier and more efficient to remove sequences from the DNA than to insert sequences. And since most of the Duchenne mutations are deletions of one or more exons, the first approach that's been explored using gene editing is to remove exons or skip exons in the DNA sequence, thus kind of recapitulating the type of approach that exon skipping uses at the RNA level, but doing this at the DNA level so that a single editing procedure would correct the DNA and then would be permanently changed and produce the new in-frame dystrophin protein. And so the work from our group and many other groups around the world initially applied this approach to use gene editing to remove exons. Either you typically shifting or skipping or deleting exons, similar to how exon 51 would be removed with exon skipping drugs, but in this case, doing it at the DNA sequence. And the model that we and many others initially used to mimic the removal of exon 51 from the human gene was the removal of exon 23 from the mouse gene. And our initial approach was to make 2 cuts around exon 23 and remove it from the genome, and thus restore the reading frame and production of a dystrophin protein. And we did this using the same adeno-associated viral vectors that are being used to deliver the microdystrophin genes. And we started in these mouse models. And just to give you an example of what those results look like, in this case, we're looking at a cross-section of muscle tissue from a normal dystrophin-expressing mouse. And when we stain for the dystrophin protein, we see that it coats all of the dystrophin fibers, doing its normal function of holding muscle together. We see in this dystrophic MDX mouse, the mouse model of Duchenne, that there's no dystrophin protein present when we do that same stain. But when we take this mouse and we inject the adeno-associated virus that encodes the CRISPR system to fix the dystrophin gene, and we wait 8 weeks and take a cross-section of that muscle tissue, now the muscle looks like this. So we've restored dystrophin protein expression in many of those muscle fibers. We can quantify that and see that almost 70% of the fibers are now expressing some dystrophin protein. And we expect that that should be sufficient in order to increase the strength of these muscle fibers. And in fact, that's the case, that when we test the ability of these muscle tissues to generate force or resist fatigue, in both cases, we see improved function of these mice compared to sham or control samples. So that general approach has been repeated in parallel and in subsequent studies by many different labs around the world. And that's very exciting for us because it shows that the general approach is general to lots of different strategies, different labs, different mouse models, different CRISPR gene editing systems, and different viral vectors. And just to give you a quick update on where some of the work that we and many other groups have done in the meantime, one of the approaches that we've been interested in is how do we restore a complete, fully functional dystrophin gene? Because as I mentioned, in all of these studies, they take a similar approach of removing exons to generate that truncated, partially functional dystrophin protein that mimics a Becker muscular dystrophy like product. However, the promise of gene editing is to be able to manipulate DNA sequence in any way. And if that's true, then we should be able to restore a fully functional gene. And so in subsequent studies, we've been interested in how do we overcome the challenges to be able to introduce new DNA into a very specific target site in the genome and thus restore full-length dystrophin. And so we've recently published a study that did this. We generated a mouse model of Duchenne that has the full-length human gene. In this case, we removed exon 52 to mimic a Duchenne-like mutation. So now this mouse that carries the human gene is missing exon 52 and the gene is out of frame. And the goal of this study is to deliver a CRISPR-Cas9 system that cuts the genomic DNA, but also delivers exon 52 and inserts it right into the spot of the genome in the dystrophin gene where exon 52 is missing. And in this study, we showed that we were able to accomplish that. The efficiency is too low right now, and we're working on ways to improve that efficiency. But we did show that it's theoretically possible to restore a full-length dystrophin gene using genome editing when one or more exons is removed. Now, the approach that we describe here would only be applicable if the patient was only missing exon 52. And that obviously would be just a very narrow segment of the patient population. And we're very interested in developing approaches that would be more generally applicable to a broader patient population. And so for that, we've explored how do we take a whole larger region of the gene and insert it into a specific site that could capture a much larger group of patients. And I won't go through the details, but this demonstrates an example where we took all of exon 52 through 79 and inserted it at one site downstream of exon 51, and that would be generally applicable to any patient who had mutations after exon 51. And that's about 20% of the patient population. But interestingly, the same approach that we have pursued here could be applied to all of exons 44 through 79 with a single AAV virus that would be applicable to about half of the patient population. And this just gives one example of one result with that study where we're actually looking at a cross-section of the heart tissue from these mice that have the human gene, where we inserted either exon 52 or all of exons 52 through 79. And you can see that we could achieve as much as 25% of the cells expressing dystrophin. The dystrophin levels are shown over here by Western blot, again, showing that either with just inserting exon 52 or the super exon that encodes all of 52 through 79, we were able to generate full-length dystrophin protein. And that approach also decreased the serum creatine kinase levels in these treated mice. And just to summarize the state of the field, I think that probably many of you have seen many studies reporting genome editing for Duchenne, and I'm sure many of you have the question of how far are we away from, and what are the roadblocks from actually being able to apply this clinically. And the work that we and others are really focused on is ensuring both safety and efficacy of this approach in humans. And I'll just talk very quickly about a couple of these issues. The first, of course, is the issue of off-target gene editing. So we know that gene editing works by changing DNA sequence. And so we are intending to change the sequence of the dystrophin gene, but it's very difficult to rule out the potential that these gene editing tools could accidentally change DNA sequence elsewhere in the genome. And so we and others are working very hard to increase the specificity of these tools to ensure that that will not happen, as well as develop new methods to be able to characterize any potential changes to the billions of base pairs in each cell that are being treated. And it's important to understand, what is the acceptable level of risk for this approach. In addition to the potential to change DNA sequence at other sites in the genome, we need to make sure that we're getting the right change at the intended target site of the dystrophin gene. And we and others have showed that even when you edit the dystrophin gene successfully, in many of the cases, you get the edit that you want, but you also can get other types of edits and other types of changes to the DNA sequence of the dystrophin gene. And so we want to make sure that we characterize that and maximize the potential to get the intended edit and minimize the unintended edits. Again, we need to understand what the acceptable level of risk is here. And then finally, there's the concern of tolerability. Some of the issues associated with safety of delivery via AAV vectors have already been well described, and I'm sure many of you have seen some of those reports. In addition to the AAV delivery vehicle, the CRISPR systems that we're using for gene editing are derived from bacteria and therefore are foreign to human patients. And so again, we're working very hard to understand how the immune system will respond to these bacterial proteins and what the acceptable level of risk is. And then finally, in terms of efficacy, we need to make sure that we can deliver to all of the tissues. We need to make sure that even once we deliver the CRISPR gene editing machinery to the tissue, that we maximize the amount of editing that happens in those tissues. And then finally, we believe that by editing the underlying DNA sequence, we can get long-term durability of the dystrophin restoration, but we need to experimentally ensure that that is the case. And we believe that many of the ongoing gene therapy studies are really informing how we can translate genome editing in the future. And so with that, I acknowledge some of the members of my lab along the way that did some of the work. And I'd like to acknowledge our collaborators and funding sources here. Again, Sarepta Therapeutics has been a really amazing partner and sponsor of this work in our group. And I look forward to the Q&A. Thank you for your time.
Grazie, professor Gesbach. Adesso andiamo al secondo intervento che rappresenta...
Thank you very much, Professor Gesbach. Let's move on to the second presentation. It's a video contribution by Professor Tedesco, a professor of neuromuscular biology and regenerative medicine at UCL London, where he runs his lab and whose main activity for years is focused on the study of muscle regeneration to study new and innovative therapies to fight neuromuscular diseases. You have the floor. Or better, let's start the video. Good afternoon, everyone, and thank you very much for inviting me to this wonderful conference. I'm really sorry I cannot take part in person because right now I'm on a plane, I'm flying, and hopefully in the next 10 minutes I can give you an idea of what my research group has been working on in our labs in London. At the UCL in the Francis Crick Institute in order to use complex models of human muscle disease in order to test new therapies and also understand or get an insight into the disease mechanisms. You need no introduction on how important muscles are and how complex they are in terms of function and structure. Let me just mention the presence of multinuclear muscle cells that contain several nuclei inside a single cell and very complex innervation and with a different complex structure and function. But the presence of many nuclei is particularly important because I'll be focusing on the role played by muscle cell nuclei. You can see these little globes that you can see inside the fibers which contain DNA. As you know, muscles are targets to many severe diseases that desperately need to be treated. So we need to work on new therapies. Our group has focused over the last few years on generating different human muscle models using alternative systems to the conventional animal model, such as dystrophic mice. We'd like to generate human muscle diseases which can be specific not only for the different diseases, but also for individual patients or specific gene mutations. So we'd like to be able to identify the early signs of disease in vitro, possibly, using culture medium in a progressive way, not only getting a snapshot of a given moment, but creating a dynamic system where we can analyze the molecular mechanisms leading to the disease and then be able to generate specific and targeted treatments. We can induce multipotent stem cells, the iPSC, indicated here, the induced pluripotent stem cells that you've certainly heard about. It is a very complex slide, which simply shows that we have a very complex system going from pluripotent stem cells, and the induced ones are generated by skin fibroblasts, which get converted into pluripotent stem cells that can then give rise to all cells in our body. By using the right protocols and cocktails, we can stipulate the muscle to differentiate. This has been done over the last few years. I've been working on it for over a decade, and there are different methods to generate muscles from iPS cells. The advantage being that we can have an unlimited amount of muscle cells that can be generated by, for instance, a skin cell. And of course, avoiding muscle cells, which would be necessary to obtain that same result. The first muscle disease model that we have studied for a number of years using iPS cells are the laminopathies. You may not be very familiar with them, but they're very severe conditions, especially when they are accompanied by congenital muscular dystrophy due to a gene called LMNA with a protein called lamin AC. They're located on the cell nucleus. They form a kind of mesh within the nuclear membrane, which performs a number of functions. I will not get into detail right now, but that promotes a structural stability and regulate gene expression. There are no treatments for this disease, and the underlying mechanism is not very clear yet. This applies to other muscle diseases such as Duchenne. Laminopathies have a particular feature when we look at this cells. The nucleus here is not round and regular, but is particularly irregular and rough-edged. Looks abnormal. Together with my colleague Pete Summit in London, we studied that to see whether our induced iPS cells that we had produced could somehow recap nuclear abnormalities. We've been working on this a lot, and we're very happy to see that where the cells differentiated into muscle tissue, they immediately showed a number of abnormalities in nuclear shape, such as these deformities that you can see here of these bubbles at the periphery of the nuclei. I will not get into details here, but here we can see that we also quantified the number of nuclear abnormalities and the muscle cells generating iPS cells. And when we went from proliferative cells, the myoblasts, to the myotubes, they are the myofibers, the muscle fibers, we saw that there was a reduction in the number of abnormal nuclei, and we would have expected the very opposite. Myofibers are usually diseased in these patients, and we would have expected to see more altered nuclei, more dysmorphic nuclei. And that what was probably due to our basic culture mechanism using a simple culture system. So there was no sophisticated system, and we thought that that might have caused this difference, unexpected difference. In order to understand what the problem was about, we took the cells I showed to you in the previous slide, and we merged the project with another ongoing project at our lab, which was based on the generation of artificial models, muscles for the iPSC-derived material. So we had a number of applications in mind, but you can see this is a general overview. One skin cell was used, and that diversified or differentiated in muscle cells, neurons, blood vessel cells, and that led to the creation of an artificial muscle with a three-dimensional structure, which very faithfully recapped the defects of an adult muscle. You can see an example of that, normal iPS cells of an healthy person. In red, this is myosin. You can see different muscle fibers with lots of nuclei and very much aligned. So this is enough for us to observe defects, as we'll be able to see in the next few slides. This is just to show you that we differentiated many different cell lineages, DMD, alpha sarcoglycan, mutation, and different types of laminopathies. In red, you can see the muscle fibers of these three-dimensional systems. This can give you a clearer idea of the 3D structure of the system. In red, in fact, particularly here, you can see dystrophin, and in green, the muscle fibers. Of course, being able to analyze the function and clearly see here that these three-dimensional systems, by using electrical stimulation, they respond by contracting. There's a muscle twitch that is totally in vitro, so it's a cell culture. The system responds to stimuli so that we can analyze the contractile force in response to different stimuli and to test different therapies. So we use these 3D systems to examine the aspects of nuclei inside the cultures, inside our medium. Surprisingly, we saw a prevalence of nuclear abnormalities. You can see this very elongated elongated nuclei. Interestingly, this kind of nuclei are objective parameters that can easily be measured by machines, by computers, and that can be used to test treatments and see whether they become more effective. Additional studies were carried out in order to see whether nuclear abnormalities was specifically observed for given types of mutations observed in the patients. we observed that the most severe mutation, indicated here, the R249RW, is the one that has the most nuclear abnormalities and the most elongated nuclei. And you can see here, in fact, that the green spots indicate the 3D muscles. They're usually always longer and higher than the gray ones, showing that the 3D system has a higher resolution, so is better able to identify nuclear abnormalities. This is even more detailed. As you can see, we can see the elongated nuclei in these patients. This is a normal control on the left. Of course, in this way, we could develop a new parameter for testing treatments in the future, and it has been recently been validated by another lab in France. What kind of therapies can be tested in this way? We started working using the exon skipping method that you know very well in the Duchenne community. It's not very much not been tested in laminopathies, for instance, and we identified a number of mutations thanks to the work carried out by Peter Zamet in the past. Unfortunately, I have no time to get into more detail, but we can use the exon skipping method to develop a treatment for laminopathies using the in vitro system and following the parameters going back to normal. So the nuclei going back to normal. This is about all I have to say. We have other projects that we're working on. It is a simple, but I hope I've been able to show that we have a simple and sensitive system to generate muscles that are specific for different muscular dystrophies. Using iPS cells, we can obtain a high-fi and high-resolution recap of particular defects of the disease, such as laminopathies. It can be done in DMD, in UCMD, and XLCNM, and in myotubular myopathy, X-linked centronuclear myopathy. Other colleagues at my university have been working on more complex systems, on muscles and endothelial cells and smooth muscle cells, vessels and motor neurons, as you can see on this picture. So by increasing tissue complexity, our theory is that we can also increase the resolution of our final recap. In cooperation with the parent project, I'd like to thank you very much for that. We're setting up an international consortium, including colleagues in Europe and the United States, where the system can be used to test a number of different complex and conventional treatments by using this in vitro three-dimensional humanized human muscle models. Thank you very much for your time. Unfortunately, I have no time to thank all my colleagues. The 3D system, however, was great work carried out at my lab by Sara Moltinetti, now at the San Raffaele Hospital, and Luca Pinton and others still have been working a lot on developing models and on nuclear abnormalities. And here we can see the list of our great colleagues and all our sponsors. Unfortunately, I won't be able to answer your questions, but I'll be happy to do that in the next few days. If you have, just let the parent project staff receive your questions, I'll be able to answer in the next few days. Thank you very much. I'd like to pick on Professor Tedesco's final message to remind all of you attending our session to please write the questions in our Q&A space, all the questions for our speakers and also the ones for Professor Tedesco, who will answer in the next few days. So please post your questions so that we can provide targeted answers.
Thank you. Well, I think we are respecting our time. So I introduce the third presentation. It is a pharmacological treatment, and our speaker is Mrs. Martina Sandona. She's a researcher at the Santa Lucia Foundation, IRSCCS in Rome. She is one of the co-workers of Professor Puria, and for many years now, she has been working in this area, trying to understand the mechanism promoting muscle regeneration. You have the floor. Thank you.
Spero vediate la mia presentazione.
I hope you can see my slides. First of all, I'd like to thank Parent Project for this opportunity and speak about our recently published project concerning the communication between two essential cell populations and how pharmacological treatment with inhibitors of the acetylase histones, so the HDAC inhibitors, can solve this situation. We have the FAPs, FAPs, and satellite cells or MUSCs, muscle stem cells. So FAPs are cells that are quiescent, that are inactive if the muscle is healthy. If you have a chronic or acute muscle damage, they activate in order to accelerate the regeneration process. In the dystrophic muscle, these cells behave as multipotent cells, so they can differentiate into a variety of phenotypes, fibroblasts or adipocytes. So in dystrophic muscles, FAPs are responsible for the deposition of fibrotic and adipose tissue. And then we have satellite cells or of the muscles that are the main actors in repairing muscle damage. They are quiescent in physiological conditions, but if there is a chronic or acute damage, they activate themselves, they proliferate, and they form this myocytes that form the new fiber, the fiber that is going to replace the damaged fiber. In a dystrophic muscle, this process of satellite cells is extremely active in the early phases of the disease, whereas it slows down during the progression of the disease. So at late stages of the disease, the fiber is no longer able to be repaired, and it is replaced by fibrotic or adipose tissue. Many years ago, in our lab, in Mr. Puri lab, we demonstrated the pharmacological treatment with epigenetic drugs that are called HDAC inhibitors is capable of fighting the normal decline of a dystrophic muscle. This drugs acting on the epigenetics, so on the environment, can increase the regenerative process and reduce the accumulation of fibrotic tissue. At the cell level, we were able to demonstrate that FAPs are also the main cell targets of this treatment. After the treatment, you have a reduction of their potential to produce fibrosis, so their ability to accumulate fibrotic and adipose tissue, and you have an increase in their myogenic potential, so their ability to form muscles. And we were able to demonstrate that in dystrophic muscles, FAPs and satellites can communicate, and FAPs communicate with satellite cells by stimulating their differentiation. This process is implemented by the pharmacological treatment with HDAC inhibitors. So moving from this state-of-the-art experiment and also the different studies on the secretoma. And when I say secretoma, I mean a number of soluble factors that are present in vesicles that are released by the cell itself. In this project, we asked ourselves what were the mediators of this communication and how the pharmacological treatment could affect this communication, and so these mediators, and so the FAPS secretoma. So we, as for the secretoma, we identify this extracellular vesicles, EVs. They are the main actors of intracellular communication, and thanks to their content in proteins, microRNA, and mRNA, they can modulate the phenotype of the receiving cells. In order to evaluate whether these extracellular vesicles had a role in our system, so the communication between FAPs and satellite cells, we inhibited the release of FAPs with a specific drug. And we observed that if these FAPs do not release extracellular vesicles, they are no longer capable of supporting the process of muscle regeneration. So these extracellular vesicles play an essential role in this process. They were also characterized from the chemical and physical viewpoints, and the characterization told us that we can isolate the smallest part of these extracellular vesicles, those that are called exosomes, and they have a range of size that is between 30 and 150 nanometers. To confirm that these vesicles were involved in this transformation, that it could transfer the beneficial effect of the treatment with HDAC inhibitors, we carried out an experiment where satellites were put in a culture with the vesicles, so not with the cells, but just with the vesicles. And we cultivated these satellite cells with these vesicles, both treated and not treated with the drug. You see, either treated or not treated, if we put the treated vesicles, there is an increase in the differentiation of satellite cells, confirming the role of vesicles in mediating this effect and the ability of transferring the beneficial effect of the drug treatment. As I've already mentioned, the content of these vesicles is extremely important. So we checked whether the drug treatment could modulate the content of these vesicles. And we concentrated on the microRNA content. So we examined the expression of microRNA in vesicles, either treated or untreated. And there is an increase in the expression of many microRNA after drug treatment, and many of these microRNA are involved in antifibrotic and muscle regeneration processes. The one that is most upregulated in these vesicles after treatment is this 206. It is muscle specific. It is described in DMD and all the process of repair of muscle damage with biotools. Well, they can act on molecules that can influence the activity of the cell. So we wanted to see whether this content was essential in their ability to lead to the differentiation of satellite cells. So we repeated the experiment that I've already explained, but in this case, we put vesicles where there was an inhibition of A206. If A206 is not present in the vesicles, there is an absolute loss of their activity, so of their ability to increase the differentiation in satellite cells. And this was observed not only in terms of cell differentiation, but also as activation and proliferation of the cells. With an experiment on the individual myofiber, we were able to see that these vesicles can expand the population of satellite cells and increase their proliferation. But if we eliminate the A206, the effect disappears. This confirms the essential role of the content of these vesicles for their activity on satellite cells. Then we checked their in vivo effect. So we transplanted these dystrophic mice with extracellular vesicles, isolated always from FAPs, but either treated or untreated. We injected them every 7 days, and we see that the vesicles isolated from treated FAPs with an epigenetic drug, they can increase the regeneration process, reduce the presence of fibrotic tissue, as well as reduce inflammation. To assess the effect that the content of these vesicles have, well, we inhibited two types of microRNA, the 206 and the 145. If a 206 is missing, we still see an anti-inflammatory effect, but we don't see the anti-fibrotic effect. If we eliminate the 145, we also lose the anti-inflammatory effect. So the vesicles isolated from FAPs can mediate the beneficial effect of the drug, and that the appropriate assembling of these vesicles is essential for their beneficial effect in fighting against the decline of the dystrophic muscle. In conclusion, with this paper, FAPs and satellites communicate through these vesicles, and that the drug treatment with HDACI completely changes the microRNA content of the vesicles, and this content becomes pro-regenerative. So it can promote the differentiation of satellite cells in vitro, and it can inhibit the decline of the dystrophic muscle in vivo. This discovery is of paramount importance because it opens up the way to a number of therapies based on the use of vesicles, which by their features can avoid all the side effects that are associated with cell therapies and pharmacological therapies. I'd like to thank Valentina Saccone and Dr. Pier Lorenzo Puri and the whole lab and all of you for listening to me. Thank you.
Thank you very much for this interesting contribution. And we'll now give the floor to the fourth and last presentation of the first scientific session of our international conference. Our last speaker is Professor Guglielmo Sorci, Professor of Human Anatomy at the Department of Experimental Medicine at the University of Perugia, with his assistant, Dr. Sara Chiappalupi. Professor Sorci and Chiara Chiappalupi have carried out a very interesting study on Sertoli cells and their anti-inflammatory potential in Duchenne patients. Project that has received funds from Parent Project, and we're very curious to know more about it. Good afternoon there, everyone, and thank you to Parent Project for giving us this opportunity. Sertoli cells are very abundant in these seminiferous tubules. They provide numerous factors required for the development of maturing germ cells and create a physical barrier. Thanks to these properties, Sertoli cells have been successfully used in different preclinical therapeutic approaches, such as neurodegenerative diseases and diabetes, both in murine models and human models. Microencapsulated porcine Sertoli cells have been encapsulated using a procedure which has been patented, developed by Giovanni Luca's group and Professor Calafiore as well from Perugia University. We've been cooperating with them for many years in developing new therapeutic and experimental treatments for DMD. We have shown that the precapsulated Sertoli cells have been inoculated into the peritoneal cavity of 4-week dystrophic mice in the acute phase of disease for 3 weeks and 5 months. After one inoculation, we observed a significant reduction of inflammatory infiltrates in the treated mice muscles. There were anti-inflammatory macrophages. We also observed that this treatment induces upregulation of trophin levels in the membranes, thanks to the release of this factor, which interacts with receptors and leads to a utrophin upregulation. This transduction pathway has been studied in human DMD microtubes, where Sertoli cells can induce utrophin upregulation independently of the mutation of the dystrophin gene. We also observed that by using another experimental model which cannot induce upregulation of utrophin, Sertoli cells induce significant reduction of muscle inflammation, showing that the anti-inflammatory effects are independent of the induction of utrophin expression. Moreover, we observed that Sertoli cells induce reduction in fibrosis in all the muscles that we analyzed in the dystrophic mice. We recently showed that in vitro treatment of dystrophic myoblasts with different mutations shows a reduction in fibrogenic markers, showing that probably Sertoli cells cause an inhibition of myofibroblast transdifferentiation. There was also a reduction in the fibrogenic potential of fibroblasts. This data can be seen in detail in a poster presented by Dr. Salvatori. Sertoli cells injected into the peritoneal cavity of dystrophic muscles induce the release of all these factors, which from the peritoneal cavity are distributed to all muscles, where they induce reduction of muscle necrosis and improvement of muscle fibrosis and fat tissue infiltrates, leading to improved muscle performance and significant damage reduction. We then looked at a number of aspects related to the safety of this treatment with Sertoli cells, and we looked at the immunomodulation induced by Sertoli cells. They can lead to the release of a number of immunomodulating factors which create a privileged environment. So far, we haven't shown whether these factors can induce immune suppression. For this reason, we asked whether they could predispose to infection or to lead to tumor development. we used two species, Aspergillus fumigatus and Candida albicans, which induce pulmonary infection and a gastrointestinal infection, respectively. In both cases, we observed that Sertoli cells, not only they do not inhibit the immune response of mice, in this case against pathogens, but actually they actually induce a rapid response to infection. As far as the tumor development is concerned, we use two different tumor lines. One, Lewis lung carcinoma, and one was murine melanoma. In both cases, Sertoli cells had no impact on tumor development. Both in the treated and control mice, we had the same tumor volume. So treated mice were not immune compromised. We also observed that Sertoli cells reduce tumor invasiveness in mice treated with pulmonary lung carcinoma cells and reduce metastasis in the lungs. Moreover, we observed that mice treated with Sertoli cells in both groups were protecting against weight loss induced by the tumor, and also prevent muscle atrophy induced by Sertoli cells. I will now give the floor to Professor Sorci for the conclusions. Good afternoon, everyone. So let me briefly recap the main advantages of the Sertoli cell-based therapeutic approach. As explained by my colleague, it is actually a drug delivery system. It's a system leading to the production of a number of anti-inflammatory effects and the expression of eutrophin, and they are circulating and reaching all muscles in the body. It is a combinational approach. We have inflammation and induction of eutrophin expression, the two being independent of one another. This approach leads to a reduction in fibrosis in the muscles, and there's a direct action on dystrophic myoblasts and direct action on fibroblasts. This approach has anti-atrophic properties for muscles affected and developing atrophy. And the anti-atrophy effect has been seen in myotubes due to a different number of stimuli. This is a universal approach, independent of the dystrophin gene mutation. And more importantly, it doesn't need any pharmacological immune suppression. One of the most important properties of Sertoli cells is that they modulate inflammation. We are convinced that this is an important discovery. We are going to continue our study. We are getting the opinion of an expert panel made available to us by Treat NMD. They recognized the effects of this therapeutic approach, and a number of recommendations have been provided on preclinical models. We need to provide additional information before we transition to humans. We have to look at the effects of the treatment on different muscles, such as the heart muscles. They need an analysis of biodistribution and pharmacodynamics of the most relevant Sertoli cell-secreted factors, we need to provide data on dosage and duration of action of engrafted Sertoli cells after inoculation. Direct measurement of muscles of treated animals. We need to provide an evaluation of subcutaneous inoculation instead of using an intraperitoneal approach. The subcutaneous injection could be easier to perform by creating a subcutaneous pouch on the abdomen, and then possible use of Sertoli cells in combination with steroids. And the toxicology studies have to be carried out, and also off-target effects have to be considered and evaluated. We're open to any cooperation with external companies in order to make this treatment with Sertoli cells as safe and useful as possible for BMD and DMD patients. Here we can see all the people that have contributed to our study. We're still working on it. We'd like to particularly thank Parent Project for supporting us and believing in our project, and Treat NMB as well for making an expert panel available to us. They provided precious suggestions and advice. Thank you very much to all of you for your attention. So, of course, we wish you good work, and hopefully your project can be as successful as we imagine it will be. So, hello, Gloria is here with us. Hello and welcome. We can now open our Q&A.
Well, now we have new questions, and maybe you can translate them for the speakers. Thank you very much indeed, Luca. And I wish to thank all our speakers for their beautiful presentations. They gave us a clear idea of all the things that are going on in the area of science to try and find the best possible way to reach a better future. We hope not too late. Of course, there will be questions about how long all this will take. We have here Gloria Antonini. She's here from the science area. And in the backstage, we also have Fernanda De Angelis, who is the third colleague. First question, good evening. I'm very glad to be here. Also, I wish to thank the speakers for respecting time limits. One question by Domenico. The question is for Professor Ganeshbach, and he's asking if the mutation or the duplication of exon 50, what could happen by using the genomic editing technique?
Yeah, so it's a great question, and I think it's maybe a general question about how gene editing can be applied to many different mutations. We get this question about picking different exons and duplications and deletions, and I would just say, in principle, any of those types of mutations could be addressable by gene editing. So, for example, if there's a duplication of an exon, you could imagine removing one of those exons. The tricky part, of course, in the case of a duplication is, ensuring that you remove only one exon and not both exons, both copies of exon 50, which would then just create a delta 50 type situation. So I think that that actually speaks to where our primary focus has been, which I didn't talk about in my presentation, but I've talked about previously, is one way to get to the largest segment of the patient population with a single gene editing approach is to remove all of 45 through 55, which, with a single gene editing step and a single composition that could, potentially gain regulatory approval, could get to about half the patient population. And so there's many different types of mutations that could be addressed by using gene editing to remove all of 45 through 55, including duplication of exon 50.
Grazie.
Grazie, molto interessante. La prossima è sempre per il dottor Gershbach.
Again, another question for Professor Gersbach. Francesca wrote the question in English, but I'm going to read it in Italian. And she says it was a very interesting talk and thanks the speaker. And the question is, I was wondering whether the approach with CRISPR-Cas9 is equally effective in transducing the different muscle fibers, type 1, type 2 fibers.
That's a fantastic question. You know, we've not looked at that carefully in my group, and I can't think of an example off the top of my head where CRISPR gene editing has been specifically evaluated in the different muscle fibers, types of muscle fibers. Of course, you're going to be limited by whatever the delivery vehicle is. So if you're using AAV9, for example, whatever fibers AAV9 gets to are going to be the same fibers that CRISPR gets to, since that's the delivery vehicle. After delivery, whether there's a difference in muscle fibers at the gene editing step, I don't know the answer to that, and I can't think of a good piece of data to point to. I don't think there should be a difference. I mean, I would point out that CRISPR gene editing has worked in basically every cell type and organism that I'm aware of, everything from plants to insects to humans and everything in between. So I think it should work in all muscle fiber types, but there may be differences that I'm not thinking of right now.
Grazie, professor Gershbach. Un'altra domanda sempre per lei.
Thank you. Another question for you. And then maybe we'll have questions also for the other speakers. And this is a question that is being asked by many parents. How long will it take? How long will it take to get to the level of a clinical trial, a trial on patients? How long are we supposed to wait?
Yeah, I mean, it's a great question, and I realize it's an important question on the front of the minds of many parents and families. And it's a very difficult question to answer, and I can't give any specific timeline. I tried to highlight very quickly at the end of my presentation where some of the gaps are that we're still trying to fill our knowledge in terms of how safe it is and how efficient it is. And I would also, I'd highlight that we're learning a lot from the microdystrophin gene therapy trials and other gene therapy trials for muscle and heart. And I think that, we're trying to incorporate learnings around safety and efficient delivery from those studies as well, which I think, continues to push the evolution of how CRISPR will be used. So unfortunately, I don't think I can give any specific time frame right now. I mean, I do believe that genome editing will be important and play a role in helping to treat genetic disease in the future. And I'll also point out that there are other diseases in the liver and ex vivo modification of stem cells and T cells where we're seeing incredible advances and success in humans in clinical trials with CRISPR gene editing. And I think we're also trying to learn from those and in order to make sure that what we do is safe and effective. And so I think there's great progress. I think the recent evidence of success in blood stem cells for sickle cell disease and gene editing in the liver with lipid nanoparticles delivering mRNA to treat amyloidosis, all incredibly exciting. And we're trying to learn from that to develop a safe and effective gene editing strategy for Duchenne.
Grazie, infatti immaginavamo che la risposta potesse...
Thank you. And we do hope that now with the trials of gene therapy that are now a reality, we hope we can learn more and more rapidly also with the other techniques, the new approach by Professor Tedesco that may lead to new solutions to test all possible problems. There is another question by an anonymous viewer and listener, and it is a question for Mrs. Sandona. How to use mesenchymal stem cells for treating DMD. Thank you. Well, our study tries to give an answer to that question because when we use mesenchymal stem cells in cell therapy, well, there are a variety of problems to reach the target organ, to keep the engraftment, the use of the secretoma and the vesicles. Well, that is still not very close to clinical trials, but we try to make it possible to use the product of MSCs in treating different pathologies, including DMD. So the use of these vesicles could be the real way to apply MSCs to dystrophy. Thank you, Martina. And now we have other questions. Can you show us the questions?
Vogliamo fare l'altra domanda, magari sui tempi?
Vado con una domanda che ci è venuta in mente ascoltando il professor.
Now a question for Dr. Sorci and Dr. Chiappalupi. I mean, we, of course, support them. We've been supporting them for some time now. All the recommendations you received, well, we have quite a few, but they can be overcome. We'd like to understand how long will it take. We know that research sometimes has slowdowns, and well, you understand very well that for patient, time is of paramount importance. Well, we have many data. So if we were to assess the feasibility, the subcutaneous administration rather than the peritoneal route, there are many parameters we already have. So we already know what are the parameters to be assessed, what are the routes that are involved, what are the serum factors that are involved. So that would accelerate things enormously. Obviously, the part pertaining to off-targets, toxicology, doses, all that requires time, money, and resources. So that's why I launched this appeal to companies that might be interested, because that would make the evolution, the development of this much more rapid. We believe in that, not only Sarah and myself, but the group that, I mean, all the people that have been working with us. Thank you. We keep fingers crossed for you. We have time for one final question. For the others, well, we'll answer them later on. We have a question by Costantino for Dr. Sandona. So the intervention of HDACI was tested only in vitro or in vivo as well. I try to understand in the phase 3 of Givinostat, we perfectly know the side effects of severe thrombocytopenia, because of which, I mean, you cannot use the dose, the required dose. They may help differentiation and regeneration of the muscles. Well, is it possible to, I mean, research has considered the aspect of tolerability in men? Thank you, Costantino. Thank you for your question. It is a complex question. Preclinical studies on mice were carried out both in vitro and in vivo with different doses. Givinostat went through clinical trial because it has already been tested in other pediatric diseases. So a number of pharmacokinetic and pharmacodynamic studies have already been assessed. And certainly the phase 2 study was useful to understand the aspects you are talking about, side effects and dose-dependent effects. As for this topic, I think Italfarmaco is going to answer in the next few days, but what I'd like to say is that our research is aimed at precisely at avoiding all these side effects. So the use of the product, of the good effect of this drug, might avoid a number of side effects. So our goal, that is precisely our goal, limiting side effects, limiting limiting the side effects and taking only the good part of the product. Well, the answer was certainly not easy. The next few days we'll go back to that. I wish to thank all the rapporteurs. And now we move to parallel sessions. There are 4 parallel sessions, so you can pick and choose. We have Easy Life Domotics for the community. We have motor and respiratory physical therapy for children and adults. And then we have another session when complexity increases, DMD and BMD and related problems, and what we eat has weight, nutritional aspects in DMD and BMD. Tomorrow we resume with a second scientific session at 10 a.m. and we'll be speaking about the scientific projects being funded by Parent Project. Thank you for listening to us. We thank Luca Genovese and Gloria and Fernanda, who is behind the scenes. Thank you.